Hello, my name is Aisosa Omere, and I'm a third-year PhD candidate in Dr. Bournomo's lab here at UTA. Today, I'll be discussing with you Edmund sequencing evolved precise native peptide C-terminal ligation. So when we talk about proteins, we first must discuss the central dogma. That is that DNA gets transcribed into mRNA, which is then translated into proteins. The protein level is the functional part of DNA. So our DNA encodes the genes that then are expressed as proteins, and different disease states can alter what these proteins look like. And so when we discuss proteoforms are the protein after all of the necessary post-translational modifications have occurred. And so when we talk about proteomics, we're looking at the total protein content within the system. In this case, we're looking at single molecule detection within a single cell. And so in proteomics, I'll be discussing mostly the kind of gold standard right now, which is liquid chromatography, tandem mass spectrometry. Within that sort of realm, there are two main areas of proteomics. There's bottom-up proteomics, which is state of the art right now, that we can take a protein or our proteoforms, we can subject it to some sort of protease digestion, which will cut up our proteins into smaller pieces that we can then analyze with LC-MS, and then identify the peptides and infer protein sequences through different computational softwares. Top-down proteomics is kind of a step up from that, where we take the protein or the proteoforms and separate them by liquid chromatography and then analyze the intact protein by tandem mass spectrometry, that we can then identify the proteoforms within the system with computational software. Despite both of these being sort of the state of the art, the disadvantage is that it costs a lot of money to get these instruments. Mass specs are not cheap, and they're relatively low throughput when we consider the fact that, especially in bottom-up proteomics, we lose a lot of the sequence information and we lose the contextual integrity of our specific post-translational modifications. We also require a large sample size to get the single molecule resolution that we desire. So chemoproteomic strategies may be the solution. So when we talk about chemoproteomics, we talk about taking a peptide and immobilizing it onto a surface, subjecting it to some sort of chemical modification, and then doing an Edmond degradation, which is a reaction in which we cleave the N-terminal amino acid sequentially and get some sort of a readout. So Edmond fluorescein sequencing right now is like the hot ticket item, and we take our immobilized peptide and subject it to a chemical modification that has some sort of fluorophore attached to it. We then do the Edmond degradation and we get a readout with fluorescence. The problem with Edmond fluorescein sequencing, though, is that there's a limited total signal for single molecule detection, limited modifiable residues for detection, and limited modification types that have only been demonstrated with synthetic peptides. So we seek to optimize by taking an immobilized peptide and subjecting it to a new kind of modification. We're looking at using metal barcodes that are attached to chemoselective probes that can modify specific amino acids. We'll incorporate our isothiocyanate, which is important for the Edmond degradation, with a specific barcode that allows us to identify a cycle number. From there, we can do the Edmond degradation and computationally structure our peptide based on the sequence that we get. This is an alteration to Edmond fluorescein sequencing that allows detection at a single cellular level. It also improves the throughput and it adds multiplexing capabilities, meaning we're able to run a lot of reactions at one time. As far as the scope of this presentation is concerned, we'll be looking more at the foundational aspect. That is the immobilization, so starting point. C-terminal mobilization is a type of enrichment strategy for Edmond-based sequencing. The current state-of-the-art uses either unnatural amino acids, so a synthetic amino acid containing an alkyne or an azide, to facilitate the adherence of the peptide to a surface, or the results are generally low yielding. And so what I've done is I've taken a chemoselective probe, diphenyl diazomethane. It reacts with carboxylic acids. We decided that this would be a good thing to attempt to target the C-terminus. So when we talk about the C-terminal reactivity, we have to consider that there are other carboxylic acids within a protein sample. And so when we see a model peptide here, we see at the N-terminus, we have a carboxylic acid. This is aspartic acid. It has a pKa, so that's the negative log of the acid dissociation constant, of around 3.0 to 4.2, compared to the C-terminus, which has a pKa of about 1.7 to 2.4, depending on its microenvironment. And so when we look at utilizing our chemoselective probe, we have to recognize that these are both acids and that these are both going to react very similarly. We've realized by strictly controlling the pH, we can selectively modify either the side chain or the carboxylic acid residues within a peptide. So Our initial immobilization strategy, we took a peptide and we used our pH control that we have established, and we saw side chain specific labeling. So this acted as sort of a protecting group, which left our C terminus completely free. We then used propargylamine, so an amine with an alkyne group and a carbodiamide mediated amide coupling to get the propargyl group onto the peptide. This allowed us to do a copper catalyzed azide alkyne cycloaddition to immobilize the alkyne onto a magnetic bead whose surface was functionalized with azides. And while this was very successful and we confirmed with fluorescence, we decided that we wanted to optimize the strategy. And so this led to the development of our C tags. C tags are C-terminal activity groups. So these are probes that possess an alkyne on them, and using our pH control, we're able to selectively modify just the C terminus. And so with our C tags at pH 2, we're able to selectively modify the C terminus. And so once that happens, we get our modified peptide that can undergo the copper catalyzed azide alkyne cycloaddition directly onto the magnetic bead surface. So this is different from our previous strategy that was 3 steps. Now this is a 2-step process. And so we confirmed the results of this reaction using fluorimetry. And so we took our modified peptide and subjected it to copper catalyzed azide alkyne cycloaddition, and we utilized fluorescein isothiocyanate, which is an N-terminal specific fluorophore. So this was more selective than previous dyes that we have tried to use. This helped to confirm the immobilization. So here we have just the beads. There's nothing on them. We just treated them with the fluorophore. And here we see our immobilized peptide with the fluorophore attached to them. We see a significant difference in fluorescence, and we see that we have a p-value of 0.000004, which signifies that it is a statistically significant number. And so with this, we hope to continue to develop more chemoselective probes and eventually be able to sequence entire peptides and eventually proteins so that we can better understand proteins and disease-specific proteoforms. Thank you.